Good morning, everyone. So today is the last class for this course. The last, the last three chapters are left. So I will start with the chapters. Okay. So the 11th chapter is pharmacological studies and herbal formulations. So first, the introduction to this chapter. The present unit deals with the study of adulterations in herbal drugs, methods for their evaluations, herbal formulations, and herbal drug reactions or interactions. Herbal medicine is the oldest form of healthcare known to mankind, used by all cultures throughout the history and throughout the world. So, what are the objectives of this unit? To describe drug alterations, their reasons and drawbacks, explain the drug evaluation, and list various drug formulations, and discuss herbal drug interactions and their mechanisms. So, what is adulteration of any drug? Let's first understand the meaning of drug. Drug may be defined as an article recognized in an official compendia, that is Indian Pharmacopoeia, National Formulary of India, intended for use in diagnosis, cure, mitigation, prevention, or treatment of diseases in humans or other animals, or intended to alter a bodily function or structure of humans or other animals. So drug can be any substance which is used to treat, mitigate, diagnose, or prevent any disease in the humans. So what is an adulterated drug? An adulterated drug means one which does not conform to the official requirements. Adulteration involves incorporation of impurities, spoilage, deterioration, admixture, sophistication, and substitution. Adulteration can be accidental or intentional. So, any substance which is not beneficial or which is not required in any drug and it is added in the drug to just increase the volume of the drug, it is called as adulteration. Reasons for adulteration. There are number of reasons for adulteration. Some are listed below. For collection, for imperfect preparation, for incorrect storage. So the quality, value, or usefulness of a drug may be affected or destroyed by the action of microorganisms, insects, and rodents due to improper storage of drug material. Factors such as temperature, moisture, and sunlight may also affect the proper storage of these drugs after adulteration. Now, what are the types of deliberate adulteration? Generally, there are five types of deliberate adulteration which makes the drugs inferior or of lower quality, such as confusion of common vernacular language or common vernacular nomenclature, cross substitution with different plant material, substitution with artificially manufactured substances, substitution with exhausted drugs, and addition of foreign matter. So it can be of five types. First one is confusion of common vernacular nomenclature. So if there is two drugs having similar name, that can be confused with other name, and we can deliberately add one drug in another drug because they are similar sounding in name. Then cross substitution with different plant material. In place of genuine drugs, substituted products of same appearance are available in market. So if their appearance is same, we can by mistake or intentionally add any other drug or any other substance in the useful drug which has to be used, and this can be substituted with some other lower inferior quality substances. Then substitution with artificially manufactured substances. Some substances resemble original drugs are artificially manufactured and are used as substitutes. Then substitution with exhausted drugs, sometimes exhausted material, that is after isolation of active constituents, may be used entirely or in a part of as substitute for genuine drug. Or addition of foreign matter, mixing of adventitious matters or naturally occurring materials with drug in excessive amount or parts of plant other than which constitute the drugs can be called as addition of foreign matter. Now, how to do a drug evaluation? Drug evaluation means confirmation of its quality and determination of its quality and purity and deterioration of nature of adulteration, determination of nature of adulteration, such as biochemical variation in a drug, determination due to treatment or storage, and substitution and adulteration. Some following techniques are involved in standardization or evaluation of crude drugs: morphological or organoleptic evaluation, microscopic evaluation, chemical evaluation, physical evaluation, or biological evaluation. Now, morphological or organoleptic evaluation. So, it can be evaluated, the drugs can be evaluated by their color, odor, taste, size, shape, and special features like touch and texture, etc. That will be the morphological or organoleptic evaluation. Then microscopic evaluation. In the identification of crude drugs, it is often necessary to examine the anatomical structure of that drugs. This involves the microscopical examinations of sections of material. In some drugs, transverse and longitudinal sections are taken and arrangement of tissue is observed. Then, chemical evaluation. Determination of active constituents in a drug by chemical process is referred to as chemical evaluation. Thus, chemical evaluation can be used as method for the analysis of active constituents. It comprises of different chemical tests and chemical assays. The isolation, purification, and identification of active constituents are chemical methods of evaluation. So there can be various chemical methods to evaluate the quality or the constituent in the drugs. Chemical evaluation also covers procurement of raw material and quality control, extraction, purification, and characterization of constituents of pharmaceutical interest, investigation of biosynthetic pathways, and quantitative estimation of that particular drug. Then physical evaluation. Evaluation of drugs on the basis of their physical properties, important physical properties of active constituents is known as physical evaluation. So it can be evaluated on the basis of moisture content, specific gravity, density, optical rotation, refractive index, or melting point. Biological evaluation. The drugs which cannot be assayed satisfactorily by chemical or physical means are evaluated by biological methods. Thus, potency of crude drug or its preparation is determined by means of its effects on living organisms like bacteria, fungi, animal tissue, or entire animal, and it is called as bioassay. This method is done for conformity of therapeutic activity of raw material or finished product. Then herbal drug formulations. WHO or World Health Organization currently encourages, recommends, and promotes traditional herbal remedies in national healthcare programs because such drugs are easily available at low cost. These are comparatively safe and people have faith in such remedies. According to study, 70 to 80% of people living in rural areas are dependent on herbal medicine for the treatment of day-to-day diseases. According to one estimate, more than 700 mono and polyherbal preparations in the form of tincture, tablets, and capsules from more than 100 plants are in clinical use. So there are around 700 drugs from 100 plants which are used clinically in various areas of the world. Ayurveda, Unani, and traditional Chinese medicine are the important system of medicines largely based on medicinal plants. The general public's use of herbal drugs has increased over the past 10 years. Now, classification of herbal formulations. Herbal formulations are classified broadly as herbal medicines and herbal cosmetics. Herbal medicines, the herbs are considered to be medicinally important if they possess the pharmacological activities of possible therapeutic use by that drug. The use of herb is varied as naturopathic medicines, Unani, Siddha, and homeopathic systems of medicines which come under Ayush department of our country also. Herbal cosmetics. The substances specially prepared to enhance beauty and increase the attractiveness of a person are known as cosmetics. Now, herbal cosmetics can be further classified on the basis of dosage forms or part or organ of the body to be applied. According to dosage forms, it can be called as emulsions, powders, cakes, oils, mucilage, jellies, suspensions, paste, soaps, and solutions. And according to part of the body to be applied, it can be classified for skins, for hairs, for nails, or for teeth and mouth, and borderline preparations. Some herbal formulations are given in Appendix 1 in the end of this unit. So you can read from that. Now, herbal drug interaction. Almost a century has passed and it has witnessed limitations of allopathic system of medicine. Lately, herbal medicine has gained momentum with the evidence that it has no side effects as such. Ayurveda, Unani, and homeopathy medicine are important systems of medicines largely based on medicinal plants. However, the prevalence of clinically significant interactions between herbals and medications is not known. A factor that may account for lack of data on the true prevalence of herbal drug interaction is that the following: a lack of information concerning the contents of the herbal products, or incomplete or inaccurate product information, or multiple ingredients in that herbal drug product. Now, overview of drug interaction. Drug interaction refers to an alteration of the effect of one drug caused by the presence of second drug. So, if there are two drugs and they interact positively or negatively, that is called as drug-drug interaction. Drug nutrient interaction similarly refers to the alteration of the effect of drug or nutrient caused by the presence of second agent. Drug interaction may be beneficial or detrimental or non-beneficial. Drug interaction, first one, drug-drug interaction occurs when two or more drugs react with each other. Drug, food, or beverage interaction results from drugs reacting with foods or beverages. And drug condition interactions may occur when an existing medical condition may make certain drugs potentially harmful. Now, mechanism of herbal drug interaction. The interaction between pharmacologically active botanicals and drug involve the same pharmacokinetic and pharmacodynamic mechanism as drug-drug interaction. So there are different pharmacologically active botanicals and drug which are involved in the same pharmacokinetic and pharmacodynamic mechanism as drug-drug interaction. Pharmacokinetic interaction may involve alteration in absorption, distribution, metabolism, or excretion of the drug. So pharmacokinetic reactions means interactions means whatever the body does to the drug, whatever the body does to the drug, such as absorption, distribution, metabolism, and excretion of the drug. Now, what is pharmacodynamic interaction? Pharmacodynamic interaction, on the other hand, is named after the relationship between the drug concentration and pharmacological response for a drug or botanical. So, pharmacodynamic interaction, on the other hand, is what the drug does to the body. So there are two parts of pharmacology, that is pharmacokinetics and pharmacodynamics. Pharmacokinetics is what the body does to the drug, means its absorption, distribution, metabolism or excretion. And pharmacodynamic interactions means what the drug does to the body, means whatever the pharmacological actions are happening in the body due to drug is called as pharmacodynamic interaction. Now, adverse drug reactions. Adverse type of reactions are classified as type A and type B. Type A or augmented reactions are normal pharmacological effects of the drug exaggerated to the point of being undesirable or intolerable for patients. Example of drugs indicated type A reactions include warfarin or heparin, which cause bruising. Type B reactions have more severe adverse effects associated with the unknown pharmacologic action of the drug or allergic reactions. Example of anaphylactic reaction to penicillin. So someone who is taking penicillin can have anaphylaxis or severe allergical reactions in their body. That type of severe immunological reactions is called as type B reactions. Now there are three types of drug interactions: drug-drug interactions, drug-nutrient interactions, or herbal drug interactions. What is drug-drug interactions? Can be Drug-drug interactions can be defined as the modulation of the pharmacological activity of one drug by the prior or concomitant administration of another drug. So if one drug is added to another drug, or when one drug is taken with another drug, if they interact while present in the body, it is called as drug-drug interaction. Now, what is drug nutrient interaction? When drugs are administered along with foods, the drug interaction may occur and can modify the activity of the drug or impair the nutritional benefits of the food. So, if any drug is taken with any nutrient and if they react in the body for positive or any negative effect, it is called as drug nutrient interaction. Herbal drug interaction. These types of interaction take place when allopathic drugs are administered with herbal drugs. So when allopathic drugs are taken with Ayurvedic drugs and if they react in our body, it is called as herbal drug interactions. So there are different types of interactions with different herbs such as ephedra, ginseng, licorice, St. John's wort, valerian. Now, there are different interactions by different allopathic medicines such as alprazolam with kava, amantadine with quinine, estimizole with grapefruit or quinine, buspirone with grapefruit, calcium channel blockers, carbamazepine, cyclosporin with grapefruit, digoxin with quinine, licorice, estrogen with grapefruit or herbal tea, lithium with herbs with diuretic properties, midazolam with grapefruit, phenelzine with ginseng, phenobarbital with quinine, quinidine with grapefruit, spironolactone with licorice, theophylline with St. John's wort, triazolam with grapefruit, dashan, ginkgo, ginseng, licorice, alfalfa, vitamin A with warfarin, ginger and quinine with warfarin. Now this chapter is over. We'll continue to the next chapter, that is chapter number 12. Second last chapter. Now, 12th chapter is evaluation and quality control of pharmaceuticals. What are the objectives of this unit? Understand various pharmacopoeias and their objectives. Discuss the origin of pharmacopoeias, differentiate between pharmacopoeia and formulary, differentiate between national and international pharmacopoeias, understand the pharmacopoeial monograph format, define quality control and quality assurance, understand the need of quality control of pharmaceuticals, and elaborate the various quality control tests of important dosage forms. Now, what is pharmacopoeia and formularies? These are the two terms which you should know. Pharmacopoeias are defined as authoritative treatises on drugs and preparations, their description, formulation, analytic composition, physical constants, main chemical properties used in identification, standards for strength, purity and dosage, chemical tests for determining identity and purity, etc. They are usually published under governmental jurisdiction. Whereas formularies simply list drugs or collection of formulas for the compounding of medicinal preparations. However, sometimes the term pharmacopoeia and formulary are used interchangeably. But there is a difference. Pharmacopoeia is the overall anatomical, physical, chemical, and strength and purity and dosage of the drug. The whole data for the drug is in pharmacopoeias, and formularies are just the list of drugs which are present in that pharmacopoeia. Each country has its separate sets of regulations pertaining to drugs and related substances which are mentioned in pharmacopoeia of the country. A few pharmacopoeias worth mentioning include Indian Pharmacopoeia, United States Pharmacopoeia, British Pharmacopoeia, European Pharmacopoeia, Chinese Pharmacopoeia, Japanese Pharmacopoeia, and so on. Overview. So, overview of Indian Pharmacopoeia. As per Drugs and Cosmetic Act 1940, the Indian Pharmacopoeia is the legally recognized book of standards for the quality of drug substances and preparations included therein. The first Indian Pharmacopoeia was published in the year 1955. Indian Pharmacopoeia Commission, IPC. The IPC is committed to the promotion of highest standards for drugs for use in prevention and treatment of diseases in human beings and animals, keeping in view the special features of the pharmaceutical industry in India. So it is mainly concerned with the pharmacopoeial composition of various drugs in the India. The overall objective of IPC has been creation of a compilation of standards that reflect the state of industry in the country and the production and testing capabilities of different units. Indian Pharmacopoeia monograph format. First, there will be description of that drug, then identification test, including infrared spectrophotometry, non-specific purity tests, impurities, both related substance and impurities that may be present as a result of method of manufacture or from degradation on storage, other tests, impurities other than related substances, and other tests such as loss on drying or water, assays and limit calculated on the anhydrous, dried or solvent-free basis to the extent possible for assay a robust and precise, though not specific method is given rather than a specific stability indicating and complex one. In the case of non-specific assay methods, control of impurities is provided separately by means of specific impurity tests. Each monograph is designed to be considered as a whole and should provide a reliable basis for judging and quality of drug substances. Now, overview of United States Pharmacopoeia and National Formulary. The USPNF is a book of public pharmacopoeial standards. It contains standards for medicines, dosage forms, drug substances, excipients, medicinal devices, and dietary supplements. It is best known national pharmacopoeia in the world, authorized by the government of United States and published by a non-government non-profit organization. Monographs and general chapters. The USPNF is a single volume combination of two official compendia, the United States Pharmacopoeias and the National Formulary. Official recognition. The US Federal Food, Drug and Cosmetic Acts designate the USPNF as the official compendia for drugs marketed in the United States. The USP monograph is given under the following headings. Official title, descriptive information including graphic formula, chemical formula, molecular weight, chemical names and chemical abstracts, registry number. Then essay, content, tolerance limits, packaging and storage conditions, USP reference standards, identification methods and analytical tests, melting range of that drug, residual water limits and determination tests, residue on ignition or the purity test, tests for determining chloride, sulfate, sulfide with limits, and limit tests determining heavy metals and organic volatile impurities. Now, overview of British Pharmacopoeia. The contents and volumes of British Pharmacopoeia is as follows. The current edition of British Pharmacopoeia comprises 6 volumes, which contain nearly 3,000 monographs for drug substances, excipients, and formulated preparations, together with supporting general notices, appendices, and reference spectra used in the practice of medicine, all comprehensively indexed and cross-referenced for easy reference. So it has 6 volumes. Different volumes contain different data. Other national pharmacopoeias. There are national pharmacopoeias like European Union, Indian and British pharmacopoeias. All the pharmacopoeias which are issued under the authority of government and their instructions have the force of law in their respective territories are referred to as national pharmacopoeias. So there are different pharmacopoeias of China, Egypt, India, Japan, Russian Federation, United Kingdom, USA, and other countries also have different pharmacopoeias. International pharmacopoeias. Developing countries not having a national pharmacopoeia have adopted one of the another countries or countries, in some cases the international pharmacopoeia, which was put forward by World Health Organization in 1951 as a recommendation aimed at minimizing or eliminating variations among national pharmacopoeial standards in different nations. The International Pharmacopoeia is published by World Health Organization, Geneva, by the WHO Expert Committee on Specifications for Pharmaceutical Preparations. Now, quality control of pharmaceuticals. The concept of quality control started off with an understanding that endpoint quality testing of a product unit is insufficient to assure its quality. Rather, the quality is required to be assured at each step of the manufacturing process so that quality is built into the product. So quality control maintains this fact that the quality of the drug should not be tested at the end product of the drug manufacturing, but it should be required at each step of the manufacturing process so that the quality of the product or the drug is built upon. Quality is the totality of features and characteristics of product or service which bear on its ability to satisfy stated or implied needs. The term quality control comprises of two words, quality and control. Control is meeting standards or the processes through which we establish and meet standards. And quality is not just confined to laboratory operations, but must be involved in all decisions concerning the quality of the product. What is quality assurance? In the European Union guidelines, a quality assurance is defined as wide-ranging concept that covers all matters which individually or collectively influence the quality of a product. Inside an organization, quality assurance provides a management tool. The quality assurance provides confidence for the customer, whether that is a pharmacist, doctor, or a patient, in the quality of the drug being supplied. An important part of systematic actions is the availability of complete system of standard operating procedures relating to that drug or any product. Hence, although there is a specific department with company called quality assurance, the achievement of quality assurance is not the duty of this single organizational unit alone, but is the responsibility of all staff members who can influence product quality in any way. Now, different quality control tests for different pharmacological dosage forms, such as quality control test tablets with their general appearance, weight uniformity, hardness, thickness, friability, disintegration of the tablet. Then, quality control of test capsules by uniformity of the weight of that capsule, drug content uniformity, disintegration. Then quality control test for parenteral or injectable dosage forms. Parenteral literally means aside from gastrointestinal tract. So any drug which is not taken orally is called as parenteral drug. It is tested by its sterility test, leaker test, clarity test, pyrogen test. Pyrogen means anything that can cause fever. Then quality control of test emulsions by determination of particle size and particle size distribution, determination of viscosity, determination of phase separation, determination of electrophoretic properties, and quality control for test suspensions by sedimentation volume, degree of fluctuation, degree of flocculation. Rheological consideration. Rheology is the study of the flow of matter, mainly liquids to MR. Micromeretic methods. Then suppositories. Suppositories can be examined by visual examination, weight variation test, hardness of suppositories or breaking test, melting range test, liquefaction time, quality control test of ointments by penetration property, absorption of medicament into bloodstream, irritant effect of that ointment. Okay, this chapter is also over. Now we come to the last chapter of this course of Introduction to Anatomy, Physiology and Pharmaceutical Chemistry. That is chapter number 13. This is the last chapter of our course. Good laboratory practices, good manufacturing practices, and good clinical practices. These three we have to study in this chapter. After studying this unit, you should be able to explain principles of good laboratory practices, good manufacturing practices, and good clinical practices. Understand the responsibilities of quality assurance program. Explain the functions of good laboratory practices authority and GLP inspectors. Describe the features of aseptic area in good manufacturing practices. Discuss the role of personnel in GMP. Explain the importance of good clinical practices. Understand different phases of clinical trials. describe the importance of Declaration of Helsinki and Schedule Y, and understand Institutional Review Board or the Independent Ethics Committee on or Institutional Ethics Committee. First, good laboratory practices. A number of countries require manufacturers or pharmaceuticals, veterinary drugs, pesticides, cosmetics, processed food products, feed additives, industrial chemicals, etc., to establish that use of these products do not pose any hazards to human health and the environment. There are different pharmaceutical products and other cosmetics which are used for betterment of human health and not any hazards to human health or environment also. Non-hazardous nature needs to be established through studies and data, which will be examined by regulatory authorities of concerned countries. Good laboratory practices is a system which has been evolved by Organizations for Economic Cooperation and Development (OECD) used for achieving the above goals. So these are different organizations which are present in each country for good laboratory practices and which confirms that the quality of the drug is maintained. To avoid different schemes of implementation that could impede international trade in chemicals, OECD member countries have pursued international harmonization of test methods and good laboratory practices. The National Good Laboratory Practices Program functions through an apex body which consists of the following members: Chairman, who can be a Secretary, Department of Science Technology, Ministry of Science Technology under Government of India, then are different members in the GLP program, different secretaries under different ministries. Now, what is definition and scope? Good laboratory practices is a system of controls and management for laboratories and research organizations to ensure the consistency and reliability of results as outlined in the OECD principles of GLP and national regulations. Good laboratory practice embodies a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, recorded, reported, and archived. Powers and functions of GLP Authority. The main functions of GLP Authority are monitor the progress of program implementation, established national GLP compliance monitoring system for test facilities on the basis of OECD principles of good laboratory practices, grant GLP certification to the test facilities based on their compliance to the OECD principles of good laboratory practice and OECD test guidelines, suspend, withdraw, and or terminate GLP certification from its certified test facilities and or may even inform relevant GLP compliance monitoring authorities belonging to OECD member should there be a need. Constitute such other technical committees or the working groups which it deems fit to complete a particular cause or need or activity, approve the rules and procedures that may be formulated for the smooth functioning of the program, technical committee and working groups, ensure that National GLP Compliance Monitoring Authority operates its systems in accordance with current OECD Council norms, maintain its international compatibility and mutual recognition, and organize and conduct scheduled, unscheduled inspections for its GLP certified laboratories. Nature of the National GLP program. GLP certification is voluntary, which undertakes such studies either for its own purpose or for others which will be eligible to seek GLP certification. It will establish and continually update a network of GLP certified laboratories. GLP certification is valid for a period of 3 years. And then again, you will have to apply for its certification for 3 years following the 3 years. GLP inspectors. The GLP program has opted in its system to impanel its inspectors, the experts who are currently employed with government test facilities, organizations, and whose qualifications experience are meeting those prescribed by the technical committee. GLP principles and requirements. Test facility organization and personnel. Each test facility management should ensure the principles of GLP are complied with in its test facility and ensure that a sufficient number of qualified personnel, appropriate facilities, equipment, and materials are available for timely and proper conduct of the study, and a proper record is maintained for every conduct which is being done in that facility. Quality assurance program. The test facility should have a documented quality assurance program to assure that studies performed are in compliance with the principles of GLP. Then facilities. The test facilities should be of suitable size, construction, and location to meet the requirements of the study and to minimize disturbances that would interfere with the validity of the study. Apparatus, material, and reagents. Apparatus including validated computerized systems used for the generation, storage, and retrieval of data should be adequate capacity, and apparatus used in a study should be periodically inspected, cleaned, maintained, and calibrated according to standard operating procedures. Test systems. Apparatus used for the generation of physical chemical data should be suitable, located, and of appropriate design and adequate capacity. The integrity of the physical chemical test systems should be ensured. Test and reference items. Records including test item and reference item characterization, date of receipt, expiry date, quantities received and used in studies should be maintained. Handling, sampling, and storage procedures should be identified in order that the homogeneity and stability are assured to the degree possible and contamination or mix-up are precluded. Standard operating procedures. A test facility should have written standard operating procedures approved by test facility management that are intended to ensure the quality and integrity of the data generated by that test facility. Performance of the study. For each study, a written plan should exist prior to the initiation of the study. The study plan should be approved by dated signature of the study director and verified for GLP compliance by quality assurance personnel as specified. Reporting of the study results. Final report should be prepared for each study. In the case of short-term studies, a standardized final report accompanied by a study-specific extension may be prepared. Storage and retention of record and materials. All the required documents related to quality assurance programs, personnel, etc. should be retained in the archives for the period specified by the appropriate authorities. Now, good manufacturing practices. The second part is good manufacturing practices, which is a term that is recognized worldwide for the control and management of manufacturing and quality control testing of food and pharmaceutical products. Good manufacturing practices takes the holistic approach of regulating the manufacturing process and laboratory testing during the process itself. GMP gives much significance for the documentation of every aspect of the process, activities, and operations involved with drug and medical devices manufacture. Location and surroundings. The pharmaceutical manufacturing unit shall be situated in a clean and tidy environment, free from any type of contamination, including external environmental, including open sewage, drain, public laboratory, etc. The building for the manufacturing unit shall be designed, constructed, adapted, and maintained to suit the manufacturing operations so as to permit production of drugs under hygienic conditions and to avoid various hazards, including entry of insects, pests, or rodents, and mix up between different categories of drugs and raw materials. So, proper location and surroundings is a must for maintaining the quality of the pharmaceutical product being manufactured in that particular manufacturing unit. Water system. There shall be a validated system for treatment of water drawn from own or any other source to render it potable in accordance with standards specified by the Bureau of Indian Standards or local municipality, as the case may be, so as to produce purified water conforming it to pharmacopoeial specification. Purified water so produced shall be only used for all operations except washing and cleaning operations where potable water may be used. So proper water system is a must for proper standard maintenance of the drugs being manufactured. Then warehousing area. Warehousing areas shall be designed and adapted to ensure good storage conditions. They shall be clean, dry, and maintained with acceptable temperature limits where special storage conditions are required. Example, temperature, humidity. Storage areas shall have appropriate housekeeping and rodent pests and vermin control procedures and records are to be maintained. Production area. The production area shall be designed to allow the production of a scientific and professional manner. There shall be provision for attracting the risk of cross contamination. Self-contained facilities shall be made available for the production of sensitive pharmaceutical products like penicillin or biological preparations with live microorganisms. Ancillary areas or the other areas. Rest and refreshment rooms shall be separate from other areas. There, these areas shall not lead directly to the manufacturing and storage areas. Facilities for changing, storing clothes, and for washing and toilet purposes shall be easily accessible and adequate for the number of users. So, proper ancillary areas are also required for proper maintenance of quality of the drugs which are being manufactured. Quality control area, which is very important. The design of the laboratory shall take into account the suitability of construction materials and ventilation. Separate air handling units and other requirements shall be provided for biological, microbiological, and radioisotopes testing area. The laboratory shall be provided with regular supply of water of appropriate quality for cleaning and testing purposes. Quality control laboratories shall be independent of production areas. Separate areas shall be provided for physico-chemical, biological, microbiological, or radioisotope analysis. Personnel. Prior to employment, all personnel shall undergo medical examination, including eye examination, and shall be free from tuberculosis or any other respiratory infection, skin, and other communicable or contagious disease, so that they don't infect other persons and contaminate the drug being manufactured, which is being manufactured in that particular unit. Thereafter, they should be medically examined periodically at least once a year so that they do not contaminate each other and contaminate the drug being manufactured. Raw materials. The license shall keep an inventory of all raw materials to be used at any stage of manufacture of drugs and maintain records as per Schedule U. All incoming materials shall be quarantined immediately after receipt or processing. All materials shall be stored under appropriate conditions and in an orderly fashion to permit batch segregation and stock rotation by a first in, first expiry, first out principle. So first in, first expiry, first out principle. Whichever drug is being made first should be first out, or whichever drug is is having the first expiry should be first out for delivery to the other patients. Equipment. Equipment shall be located, designed, constructed, adapted, and maintained to suit the operations to be carried out. The layout and design of the equipment shall aim to minimize the risk of errors and permit effective cleaning and maintenance in order to avoid cross contamination, buildup of dust or dirt, and in general, any adverse effect on the quality of the products. Documentation and records. Documentation is an essential part of the quality assurance system and as such shall be related to all aspects of good manufacturing practices. It aims to define the specifications for all materials, method of manufacturing, and control to ensure that all personnel concerned with manufacturing know the information necessary to decide whether or not to release a bath of drug for sale and to provide an audit trail that shall permit investigation of the history of any suspected defective batch. Labels and other printed materials. Labels and are absolutely necessary for identification of the drugs and their use. The printing shall be done in bright colors and in a legible manner. The label shall carry all the prescribed details about the product and all containers and equipment shall bear appropriate labels to avoid confusion with one drug for another drug. Quality assurance. This is a wide ranging concept concerning all matters that individually or collectively influence the quality of a product. It is the totality of the arrangements made with the object of ensuring that products are of quality required for their intended usage. Quality control system. Quality control system shall be concerned with sampling, specifications, testing, documentation, release procedures which ensure that the necessary and relevant tests are actually carried and the materials are not released for use nor products released for sale or supply until their quality has been judged to be satisfactory. Product containers and closures. All containers and closures intended for use shall comply with the pharmacopoeial requirements. Suitable validated test methods, sample sizes, specifications, cleaning procedure, and sterilization procedure, wherever indicated, shall be strictly followed to ensure that these are not reactive, additive, absorptive, or leach to an extent that significantly affects the quality or purity of the drug. No second hand or used containers and closures shall be used. Whenever bottles are being used, the written schedule of cleaning shall be laid down and followed. Where bottles are not dried after washing, they should be rinsed with deionized water or distilled water as the case may be. Master formula records. There shall be master formula records relating to all manufacturing procedures for each product and batch size to be manufactured. These shall be prepared and endorsed by the head of the production and quality control. The master formula shall include the number of product together with product reference code relating to its specifications, the patent or proprietary name of the product along with generic name, a description of the dosage, form, strength, composition of the product, and batch size, name, quantity, and reference number of all the starting materials to be meant to be used. Mention shall be made of any substance that may disappear in the course of processing, a statement of the expected final yield and the acceptable limits and of relevant intermediate yields wherever applicable, a statement of the processing location and principal equipment to be used. The methods or reference to be methods to be used for preparing the critical equipments including cleaning, assembling, calibrating and sterilizing, detailed stepwise processing instructions and the time taken for each step, the instructions for in-process control with its limits, The requirements for storage conditions of the products, including the container, labeling, and special storage conditions wherever applicable, any special precautions to be observed, and packing details and specimen labels. Packing records. There shall be authorized packaging instructions for each product, pack size and type. A batch packaging record shall be kept for each batch or part batch process. It shall be based on the relevant parts of the packaging instructions, and the method of preparation of such records shall be designed to avoid transcription errors. Then product recalls. A prompt and effective product recall system of defective products shall be devised for timely information of all concerned stockists, wholesalers, suppliers up to the retail level within the shortest period. The license may make use of both print and electronic media in this record. Complaints and adverse reactions. All complaints thereof concerning product quality shall be carefully reviewed and recorded according to written procedures. Each complaint shall be investigated, evaluated by the designated personnel of the company, and records of investigation and remedial action taken thereof shall be maintained. Specific requirements. Sterile products being very critical and sensitive in nature, a very high degree of precautions, prevention, and preparations are needed. Apart from general requirements of good manufacturing practices for premises, materials, etc., for pharmaceutical products, some additional requirements shall be complied with the manufacturing of the sterile products, parenteral preparations, and sterile ophthalmic preparations. Aseptic areas. The sterile items have to be manufactured in aseptic conditions to make them free from microorganisms. Extra care and precautions have to be taken in the design and construction of the aseptic areas. Walls, floors, and ceiling should be impervious, non-shedding, non-flaking, and non-cracking. Flooring should be unbroken and provided with curved block, curve both at the junction between the wall and the floor as well as wall and the ceilings. Walls shall be flat and ledges and recesses shall be avoided, and ceiling shall be solid and joints shall be shielded. Light fittings and air grills shall flush with the walls and not hanging from the ceiling so as to prevent contamination. Garments. The garments required for use of personnel working in aseptic area shall be of special type. Outdoor clothing shall not be brought in the sterile areas so that it does not contaminate the area and the drug being manufactured. The garments shall be made of non-shedding and tight weave material. Cotton garments shall not be used. The garment shall shed virtually no fibers or particulate matter. The clothing and its quality shall be adapted to the process and the workplace and worn in such a way as to protect the product from contamination. Now the last topic is good clinical practices. First, the origin of concept of good clinical practices. The history of good clinical practices state traces back to one of the oldest enduring traditions in the history of medicine, that is the Hippocratic Oath. As a guide, as a guiding ethical code, the Hippocratic Oath is primarily known for its edict to do not harm to the patient. However, the complexities of modern medicine research necessitate a more elaborate set of norms and guidelines. Physician has to comply with some ethical and scientific responsibilities, such as obtaining informed consent or disclosing risk while involved in biomedical research. Definition of good clinical practice is a set of guidelines for biomedical studies which encompasses the design, conduct, termination, audit, analysis, reporting, and documentation of the studies involving human subjects. The fundamental tenet of GCP is that in research on man, the interest of science and society should be never take precedence over considerations related to the well-being of the study subject. So, whenever you are doing any study on a human subject, doing any clinical trial or any vaccine trial, there should be good clinical practices which should be maintained, such as the risk benefit ratio is balanced. If there are more risks and less benefits, then the procedure should be stopped. But if there are more benefits and few risks, it can be done with proper maintenance of standard operating procedures. Principles of GCP are as given below. Clinical trials should be conducted in accordance with the ethical principles that have their origin in Declaration of Helsinki and the consistent and are and are consistent with GCP and the applicable regulatory requirement. Before a trial is initiated, foreseeable risks and inconvenience should be weighed against the anticipated benefit for the individual trial subject and society. A trial should be initiated and continued only if the anticipated benefits justify the risk, which I told you earlier. The rights, safety, and well being of the trial subjects are the most important considerations and should prevail over interests of science and society. The available non-clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. Clinical trials should be scientifically sound and described in a clear, detailed protocol. A trial should be conducted in compliance with the protocol that has been received prior institutional review board or institutional ethics committee approval under favorable opinion. The medical care given to and medical decision made on behalf of subjects should always be responsibility of the qualified physician or when appropriate of a qualified dentist. Each individual involved in conducting a trial should be qualified by education, training and experience to perform his or her respective tasks. Freely given informed consent should be obtained from every subject prior to clinical trial participation. All clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. The confidentiality of records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. Investigational products should be manufactured, handled, and stored in the accordance with applicable good manufacturing practices. They should be used in accordance with approved protocol. Systems with procedures that assure the quality of every aspect of trial should be implemented. Now we come to Helsinki Declaration. The Helsinki Declaration was given by the Medical Association and has developed as a statement of ethical principles to provide guidance to physicians and other participants in medical research involving human subjects. Medical research involving human subjects include research on identifiable human material or identifiable data. The declaration was originally adopted in June 1964 in Helsinki, Finland, and has since undergone 5 revisions and 2 clarifications. The World Medical Association has developed the Declaration of Helsinki as a statement of ethical principles to provide guidance to physicians and other participants in medical research involving human subjects. Medical research involving human subjects include research on identifiable human material or identifiable data. Now, clinical study or trial. Any investigation in human subjects intended to discover or verify the clinical, pharmacological, or pharmacodynamic effects of an investigational product or to identify any adverse reactions to an investigational product or to study absorption, distribution, metabolism, and excretion of an investigational product or any drug with the object of ascertaining its safety or efficacy. The terms clinical trial and clinical study are synonymous. Clinical trials on patients in different countries are approved and monitored by different regulatory agencies. In India, it is governed by Drug Controller General of India (DCGI) office through its subsidiary known as Central Drug Control Organisation. In UK, it is monitored by Medicine and Healthcare Products Regulatory Agency as advised by the Committee of Safety of Medicines, while in USA, the Food and Drug Administration is in charge of clinical trials. Now, Institutional Ethics Committee or Independent Review Board. The composition of Institutional Ethics Committee should consist of a reasonable number of members who collectively have the qualifications and experience to review and evaluate the science, medical aspects, and ethics of the proposed trial. It is recommended that it should have at least 5 members, at least one member whose primary area of interest is in non-specific area, and at least one member who is independent of institution or trial site, and also the chairman who is not the member of the trial site or trial organization. The functions and objectives of the Institutional Ethics Committee are given below. The IEC should perform its functions according to written operating procedures. It should maintain written records of its activities and minutes of its meetings, should comply with GCP and with applicable regulatory requirements. An IEC should make its decisions at announced meetings at which at least a quorum, as stipulated in its written operating procedures, is presented. Only members who participate in the IEC review and discussion should vote, provide their opinion and or advice. The investigator may provide information on any aspect of the trial but should not participate in the deliberations of IEC or in the vote or opinion of the IEC. An IEC may invite non-members with expertise in special areas for assistance. Who is the investigator in IEC? IEC is an investigator is an individual who actually conducts a clinical investigation under whose immediate direction the drug is administered or dispensed to a subject. In the event of an investigation, it is conducted by a team of individuals. The investigator is responsible leader of the team. It is not in ISC, it is in the study trial. Okay. Investigator is an individual who actually conducts the clinical trial or clinical investigation. Sponsor. Sponsor means a person who takes responsibility for and initiates a clinical investigation. The sponsor may be an individual or pharmaceutical company, governmental agency, academic institution, private organization, or other organization which takes responsibility for the initiation, management, and or financing of a clinical trial. Contract Research Organization. A contract research organization, CRO, is an organization that offers clients a wide range of pharmaceutical research services. A sponsor may transfer any or all of the sponsor's trial related duties and functions to a CRO, but the ultimate responsibility for the quality and integrity of the trial data always resides with the sponsor. The CRO should implement quality assurance and quality control. Monitoring. The purpose of trial monitoring are to verify that the rights and well-being of human subjects are protected, the reported trial data are accurate, complete, and verifiable from source documents, the conduct of the trial is in compliance with currently approved protocol or amendments with GCP and with applicable regulatory requirement. Schedule Y of Drugs and Cosmetics Act. Schedule Y deals with regulations relating to clinical trial requirements for import, manufacture, and obtaining marketing approval for a new drug in India. The procedure for applying for marketing approval depends on the status of the new drug, which can be broadly classified into three categories. New drug substances discovered which are already approved, marketed in other countries. New drug substances discovered which are not approved, marketed in other countries, and a new drug substances discovered in India. Now we have come to the end of this chapter also, and hereby we end this course of good introduction to anatomy, physiology, and pharmaceutical chemistry. Now we have come to the end. If you have any doubts, you can ask.
No, sir.
Okay. Okay, then thank you. Have a good day. And
Thank you.
my colleagues will continue with other courses in next week.
Thank you, sir.
Okay, thank you.